So in today's class, we are going to cover our two topics, which is the transporter pumps and our signaling transduction, which is the signaling molecules. So let's begin this section with transporter pumps, which are also known as ATP powered pumps. Basically, these pumps were strictly utilized as a primary active transport because they hydrolyze ATP to generate the necessary energy to force molecules or ions against the concentration gradient from any area. Okay, it means from any area of low concentration to high concentration. Now let's begin with this topic in detail. See, transporter pumps. Pumps strictly utilize primary active transport. They hydrolyze ATP to generate the necessary energy to force molecules or ions against their concentration gradient. Means their mechanism follows, unlike channels or simple carriers, they constantly work to maintain the distinct chemical environment inside the cell. If we talk about the direction of these pumps, they were against the gradient, means they uses the active transport, neither passive. So before starting your transporter pumps, let's discuss it with carrier proteins of facilitated diffusion. So if we talk about the energy requirement in transporter pumps and in your carrier protein, then yes, in transporter pumps it requires ATP, but in carrier proteins it is not required by any energy, it is energy free. And if we talk about the direction, so your transporter pump is against the concentration gradient, but your carrier proteins is within the concentration gradient. अगर इसके हम प्राइमरी मैकेनिज्म की बात करते हैं, then your transporter pump hydrolyzes ATP to change the shape and force of ions to across the membrane. But in your facilitated diffusion, it binds a molecule spontaneously and shift its shape and releases it. Basically, so from here let's begin with our transporter pumps, in which first we are having is P-type pump. So in this, it is present in plasma membrane of bacteria, plants, fungi, and in smooth endoplasmic reticulum of animals. Then it is conserved domain, or we can say it is the one domain. It is a kind of a proton pump, but it maintains 25% energy of the body, which proton pump maintains. Then a very famous example in it having is sodium potassium pump. So this is the sodium potassium pump. Basically in first step, the cytoplasmic of sodium, it binds to the sodium potassium pump. This area is your cytoplasm and this is the pump area. Then in second step, before moving to the second step, ATP is converted into ADP. Then your sodium potassium pump is phosphorylated by ATP and the pump changes its conformation causing sodium release. ठीक है, sodium, these sodiums were attached in this surface and these were released after the second step when the sodium potassium pump is phosphorylated. Then after releasing the sodium, it causes extracellular potassium binds to the pump, leading to dephosphorylation. Then here your potassium binds to the pump and which leads to dephosphorylation. After this, the pump return to its original conformation and then your potassium is also released from the pump. This is your sodium potassium pump. Before moving to second type of pump, which is F type, your P type pump in animal plasma membrane, your sodium ion pump is present but potassium is not present. वहीं पे in the stomach, your hydrogen and potassium ions were present, neither sodium ions. Then moving forward to the F type pump, it is present in plasma membrane of bacteria, chloroplast and mitochondria. But in chloroplast, it is present in the thylakoid membrane. And in mitochondria, it is present in the inner membrane of mitochondria. Then it is used in transportation of pump, and it is active transport, means against the gradient. ATP use होगी इसमें भी. And it is not ATP binding domain, ये ATP binding domain नहीं है. Then the third type of pump we are having is V type pump. V type pump, basically it is found in the membrane of vacuoles, peroxisomes and lysosomes. In the membrane of vacuole, it is present in the tonoplast membrane. And peroxisomes and lysosomes, but it is same as of p-type pump. It works as it is of p-type. Then moving forward to fourth type of transporter pump, which is the ABC transporters. Here ATP binding domain and ATP binding cassette. It is then it is found in archaebacteria, plants and animals. They are highly conserved transports, but these in cancerous cells, ABC transporters are overexpressed. They are non-gated channels or pumps. If they will be against the gradient, then ATP will be used. If it will within the gradient, then it will work through the diffusion membrane. Here we have completed our transporter pumps. Let's begin our second topic of today's session, which is the signaling transduction. So let's start our second topic, which is the signaling transduction. So the first topic is, what are the signaling molecules? Basically, they are the chemical substances, or we can say messengers, that carry information from one cell to another to trigger a specific response. Now, only those cells respond to signaling molecules that have receptor for it. It means the molecules that bind receptor are called as ligands. Suppose this is the sending cell which is sending the your signal. These were the signaling molecules. This is the your sending cell. Okay, then these signaling molecules will move to the targeted cell, which is this, and it will having its own receptor. Then these signaling molecules will bind here only. This is your targeted cell, and this is the cell, this is the non-targeted cell. It means it is having no receptor for any ligands. Now in signaling molecules, first we are having is autocrine or autoregulation signaling. What is it? Basically, it is a chemical messenger that a cell secretes to regulate its own activity. Basically, signaling molecule activate its own cell as your cancerous cells. If we talk about autocrine signaling molecule, that it is the self-targeting cell, it acquires local action and it also helps in coordination and growth. In autocrine, if this is the cell, then it will activate its own cell, means the signaling molecules will go from here and receive by the same cell, whereas in other signaling molecules the signal were transferred into the another cell or another cell of the same cell. But in your autocrine signaling molecule, the signal produced by the molecule, that molecule get attached to the cell and the signal is received, means signaling molecule activating its own cells, it acts on the same cell. Then moving forward to the second, which is the paracrine signaling molecules. In this, it is a chemical messenger secreted by a cell to induce changes in nearby or adjacent cells, means it is localizing its response, means in the same organ of the body and response will be localized. Okay, means in the same organ of the body but different cell of the same organ, means which is your paracrine signaling molecule. It travels a short distance, means it is unstable, it is localizing, it is forming to the nearby cell. Let's see it with a diagram. If this is the autocrine signaling molecule we were having, but this is the another cell of the same organ. If it is localizing its response, means its cell का response इसी पे आ रहा है, then it is the autocrine signaling molecule. But अगर इस cell का response दूसरे सेल पर आ रहा है ऑफ द सेम ऑर्गन, देन इट इज़ द पैराक्राइन सिग्नलिंग. बट वहीं पे अगर इस सेल का रिस्पॉन्स किसी दूसरे सेल पर आता है, सपोज दिस इज द अनदर सेल ऑफ द अनदर ऑर्गन, देन इट विल बी द एंडोक्राइन सिग्नलिंग मॉलिक्यूल. क्लियर है ये? So here is your third, which is the endocrine signaling molecule. Basically, endocrine signaling molecules is a hormone secreted by specialized glands into the bloodstream to act on targeted cell in the distinct parts of the body. Means they are the cluster of signaling molecules. They were released by glands in small quantity, which are known as hormones. Means signaling molecules, they travel through bloodstream or distant cells via blood, right? So, in the process of your endocrine signaling molecule, it works as secretion like your specialized endocrine cells. In the suppose in the pituitary, thyroid, or pancreas, they synthesize and release hormone directly into the bloodstream. Or after secretion, we can say that transportation also occurs. Means the hormones travels through the circulatory system. They were reaching virtually to all parts of the body. And after this, they were the targeting to the specific receptors and response clearing. If we take a example of real world, then we were having the insulin, like it is released by the pancreas when blood sugar spikes. It travels through the blood to signal your muscles and fat cells to absorb glucose from the bloodstream. And after insulin, we can go with estrogen, which is produced by the ovaries. It acts on various distant organs to support reproductive health and secondary sexual characteristics. After the endocrine signaling molecules, we are having the another one, which is the juxtacrine signaling molecules. Basically, they are the specialized cells of biological messengers that remain firmly anchored to the membrane of the signaling cells. Means they acquires physical contact between cells. They are the specialized class of biological messengers that remain firmly anchored to the membrane of the signaling cell. Means instead of being secreted to float or diffuse to distinct targets, means as your hormone signaling, these molecules initiate a response only when adjacent cells makes direct physical contact, because these cells requires contiguous membrane to membrane contact. They are often referred to as contact dependent signaling. After this, if we talk about its key characteristics and mechanism, then it is having no diffusion. There is receptor interaction. Basically, it is the membrane bound ligand which is physically locked into the target receptor or the surface of the neighboring cell. Or it is also having direct cytoplasmic exchange. But rather this, we are having its neurodevelopment. What is it? When molecules like your afferents and semaphorins, they plays critical roles in cell migration, guiding nerve fibers, and organizing the complex structure of the brain. So after these characteristics, we are having its biological importance also. Like this is the highly localized form of communication which prevents cell from mistakenly activating pathways in distinct parts of the body. Basically, it allows for strict spatial control, which is crucial during embryonic development. Means determining the cell fate and pattern formation, and for maintaining healthy tissues architecture. Those, if you are interested in embryonic development, then you will definitely be going with this juxtacrine signaling molecule in very detail. So with this we have completed our today's both topics, which is your pumps and your signaling molecules.